Welcome back. In the last two lectures, I've described how genes, composed of DNA, are expressed outwardly as the phenotype, which I defined as proteins. DNA has an information content in nucleotide bases. Protein has amino acids that allows a protein to fold and have a function. In this lecture, I'd like to describe how this happens, how information gets translated from DNA to protein. My opening story is about toxic revenge on gene expression. In 1978, Georgy Markov was a journalist waiting for a bus in London. Markov, a Bulgarian, was an expatriate who had written several articles critical of the then government in his home country. A man carrying an umbrella, not an unusual sight in London, brushed up against Markov. Markov felt a pinprick as the man brushed by, and a few hours later, Markov felt rather weak. 2 days later, he was dead. Police found a tiny pellet where the umbrella had poked Markov, and chemical analysis of this pellet revealed a poison called ricin, which is extracted from seeds of the castor bean plant. Now, I remember when I was a kid, I was given castor oil once a month by my mother, and she told me it would clean out my stomach. I hated this stuff. It was the vilest tasting thing I can recall. In fact, one of the great puzzles of my youth was listening to an ad on the radio saying, and the ad said, "Children cry for this product." I won't give you the name of the product. I couldn't figure that out. I guess they were crying because of the anticipation of having to swallow that stuff. Well, fortunately, ricin as a protein doesn't dissolve well in castor oil. Otherwise, I would have had a really good excuse to avoid the awful taste of that stuff. Since the Markov assassination in 1978, ricin has continued to be in the news. It was used by the Iraqis in their war with Iran in the 1980s. It's the partial basis of weapons of mass destruction that the government of Iraq was allegedly stockpiling prior to 2003. In 2002, some ricin was found in caves in Afghanistan that had been abandoned by Al-Qaeda. In 2004, traces of ricin were found in the mailroom of the U.S. Senate building in Washington, D.C., and the entire building had to be evacuated. This stuff is really poisonous. A 1/10,000th of an ounce can kill a person. How does ricin work? It works by inhibiting gene expression. The castor bean plant makes ricin as a storage protein in its seeds. The seed of a plant, as I will describe when we talk about agriculture in Lecture 23, is a lunchbox. It's got stored proteins and starch to provide, respectively, amino acids, which are the building blocks of proteins, and sugars, which are the building blocks of starch, for the developing embryo. So as the castor bean plant seed germinates, it sends out chemicals that will degrade or catabolize. Now we have a word I talked about in the last lecture. It breaks down ricin to its amino acids, and those amino acids can be refashioned by the embryo of the plant for their own use. Now, unfortunately, ricin has an enzymatic activity that affects animal ribosomes. And ribosomes are the place where proteins are made in the cell. This enzyme catalyzes the modification and breakage of an essential substance in this ribosome workbench for making proteins. And so when this substance is no longer present, the ribosome is no longer able to catalyze the formation of proteins. No making of proteins means no phenotype, expression of genes. Cells die, and people die too. This lecture is about translating information that is in the gene, as DNA, as its expression, phenotype, protein. And I want to talk about that in a couple of ways. First of all, I'll try to set up the problem of translation. We've got two different languages here. Secondly, I'll try to talk about information signals for making proteins, and then get to something called the genetic code, which is an amazing translating device. We have seen how genes are expressed as proteins, and this poses a big chemical problem because genes, DNA, are real different from proteins. The building blocks of DNA are chemicals that are totally different than their expression as proteins. It's kind of like saying English is a totally different language from Hebrew. The letters look different, the meaning is different. They even do it from a different direction, right? English is read from left to right, Hebrew from right to left. Proteins are composed of amino acids, carbons, hydrogens, oxygens, and they're pretty regular in their linear structure. DNA is a double helix composed of these nucleotides that have nitrogen and sugar and phosphate, a sugar-phosphate backbone with these DNA bases. And you remember what they're called? A, T, G, and C. I'll give you the names again: adenine, thymine, guanine, and cytosine, and I promise not to give you those names ever again in the course. From now on, we'll call them A, T, G, and C. And there's 20 amino acids in protein, not just 4, as there are bases. There are 20 amino acids in protein. These molecule structures look totally different. DNA is a linear double helix. It can bend slightly, but it is largely a long fiber. Protein is in 3 dimensions. And I described, when I described protein structure in a previous lecture, how this thing folds spontaneously when it's put into water because of the properties of its constituent beads on the chain. These things are even different in their location in the cell. DNA, I think I've made this point abundantly in the course, is located overwhelmingly in the nucleus of the cell. The vast proportion of DNA is in the chromosomes residing in the nucleus, these huge molecules. Proteins, well, they're all over the cell, actually. There's proteins in the nucleus, as I mentioned before. There's proteins everywhere because of their vital functions as structural elements and as enzymes. This is the phenotypic expression of genes. Now, DNA as a linear sequence and protein as a 3-dimensional sequence. Protein is made in a location that's quite distant in cellular terms from DNA. Protein is made at the ribosome, and the ribosome is a small particle outside of the nucleus, out there in the soup of the cell. Now, how do we know this? We know this because we can take a cell and feed it amino acids that are labeled in some way. Now, the label I will choose will be a radioactive label, but you can think of it as amino acids labeled with red dye. And then you ask the question, where do we get proteins being hooked together? Where does that red dye get hooked together to form proteins? So you look for the red proteins that are just being made. And if you catch a protein just in the stage of being made, that protein will be made at the ribosome, which is far away from the DNA. So right away, we have a geographical problem as well as a chemical problem. Information signals are very important in protein synthesis. And the first thing that has to happen is that DNA has to send some information and signals for protein synthesis out to the ribosome. Now, there's 2 ways to look at this. The DNA itself could go out to the ribosome. That is, the gene or chromosome that we want to express could leave the nucleus and go out to the ribosome and say, I have the information to make this particular protein, do it now. That's one way to look at it. The other way to look at it is the DNA would say, well, you know, I've got 1,000 genes per chromosome and 23 chromosomes. It would not make sense for 1,000 genes to be made at once. They're to be translated into protein at once. So if we're only going to translate or express a couple of those genes, why not just send a copy of the DNA out to the ribosome, and the DNA can stay in the nucleus? Think of an architectural firm. Supposing you are building a house. Well, you're supervising the building of a house. Okay, you're paying for the building of a house. And a contractor has assigned to build a house for you, and the contractor has architectural plans from an architectural firm. The architectural firm might be downtown in your community, and you may live in the suburbs. Does the architectural firm take the original plans, the only copy, and drive out to your work site and have those plans there as you build the house? No. That architectural firm might be supervising the building of 50 houses at that time. They all might be different, and so what they do is, of course, they make a copy. They go down to the local copy shop and they make a copy of those plans and they send it out, and the original copy remains at the architectural firm. Think of gene expression, DNA expression, in the same way. DNA is expressed as proteins, and the copy of the DNA is being sent out to the ribosome. Now, the exact chemical nature of this copy is not as DNA. It's a different molecule called RNA. RNA is, well, it's obviously a cousin of DNA because it's got an R instead of a D. D stands for deoxyribonucleic acid, DNA. R stands for ribonucleic acid. And the sugar is the big difference. There are two differences. The biggest difference is the sugar, and the sugar in RNA is ribose, sugar, whereas the sugar in DNA is deoxyribose. So they're slightly different. RNA is single-stranded. It's not a double-stranded molecule. And that's very important because if RNA is single-stranded, its sugar phosphate backbone exposes those beads to the outer world, doesn't it? Because the beads are hanging down from the chain. If DNA is a double-stranded molecule, the beads are internal, right? It's two chains with the beads hanging down, and those beads are internal inside the molecule. They're not exposed to the outside environment of the cell. So DNA and RNA are different in that way. RNA, being single-stranded, has its bases, its nucleotide bases, exposed to the outside. That's going to be very important because RNA is going to act as a message from DNA. That's why, of course, it's called messenger RNA. It's a message from DNA, says, make this protein. But the language is still in nucleotides. It's still in bases. The enzyme that catalyzes the production of RNA from DNA has a name, and like DNA polymerase, this is called RNA polymerase. And it makes a base-paired copy from DNA. Now, what do I mean by that? Let's consider the base pairs in DNA. You'll recall, A always pairs with T. G always pairs with C. Okay? T always pairs with A. C always pairs with G. I think I've got it across both ways. These bases fit into one another and recognize one another. So if we have a region of DNA that, for my hypothetical purposes, has the following sequence, A, the other strand of DNA will be what is called complementary. We'll have the bases that fit into this, will be T, 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 T. Okay? So we've got two strands of DNA, A, and T, 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 T. Now let us say that the information content is in that first strand, A. If we make an RNA copy from the opposite strand of DNA, T, 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 the opposite bases in RNA will be A's, correct? In DNA, the opposite, the A goes with T, and now the bases opposite in RNA will be A's again. So we're making a copy of that original gene, A, and that's the molecule that goes out to the ribosome. Let's step back a minute and think of the fact that DNA is double-stranded, and now think of why DNA might be double-stranded. What a marvelous molecule this is, because the double-stranded DNA allows for its duplication Each of the new strands of double strands of DNA has a complement from the old strand of DNA. And so we had semi-conservative in its replication. That's really nice. If DNA was single-stranded, it wouldn't work that way, because an AAAAA, if it was just single-stranded, would make a TTTTT. That's not AAA. You can't make A's off of A's. You have to make it by the complementary, the bases that fit each other. So, nice that DNA is double-stranded. It's nice that DNA is double-stranded for gene expression, because we can have AAAA and then TTTTTT in the other strand, and now off of that T's, you can peel off a bunch of A's and mimic the original strand. So, we have our AAAAAA going out of the nucleus out to the ribosome. How's it get there? Well, there's a lot of mechanics going on here in the cell. And people have looked at all of the minor minutiae of how things leave the nucleus. The nucleus has little holes in it through which the molecules of RNA can move, etc. I don't want to talk about that. That's just all complex machinery. But there's a really important aspect here we have to address, and that's the following. Each chromosome, piece of DNA, of the 23 chromosome pairs in a human genome has, over 100 million base pairs of DNA. This is a long molecule. We only express at a given time in a cell certain genes. We don't express all genes at all times. Give you an example. You don't make hemoglobin in your hair. You don't make hair in your bone marrow. Profound and important conclusion. Please send Nobel Prize to the following address. Not trivial, though, because we have the genes for making them. So if we consider a long chromosome that might have the genes both for making hair and hemoglobin, the protein component of hemoglobin, the question now is, what's going to cause in an immature red blood cell in your bone marrow the hemoglobin gene to be making its RNA copy, to make hemoglobin out the ribosome, and in the hair follicle to make the hair protein and not the hemoglobin protein? There has to be an information signal. And the information signal has to reside in the DNA itself. So a gene is not just a sequence of information that determines which protein is going to be made in the order of amino acids in that protein. The gene also has to have a sequence adjacent to it that says, turn me on here in the red blood cell and not in the hair. That sequence is a group of nucleotides that's different, obviously, for the hair gene than it is for the hemoglobin gene. And that sequence of nucleotides is called a promoter. This is a signaling sequence for gene expression. A promoter is a sequence of nucleotides that is recognized by proteins. Now, what do I mean by that? I've given you the impression that in terms of information content, DNA is pretty important, right? Because the A, G, T, C, 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 C has a language. But now I'm telling you that the A, G, T, and certain sequence of nucleotides has also information of a three-dimensional nature that can be recognized by proteins that fold in a three-dimensional nature. So, if we have a protein called RNA polymerase will land on DNA only at that promoter region. That's not helping us at all, because RNA polymerase will land at the promoter for hair in my bone marrow, and my bone marrow will start making hair. If RNA polymerase just lands there, it'll start making the RNA copy of every particular gene, because every gene has a promoter. Promoters also attract other proteins. and it's called a complex. It may be due to the people who study this thing psychologically, but it's a complex of proteins that land there. Let me give you an example. Hemoglobin, as I've mentioned, is made in red blood cells. There's a hormone that stimulates this. That's going to be the big difference. The hormone enters the red blood cell in the bone marrow, and the hormone binds to molecules of protein in the bone marrow, and they together go into the nucleus and bind to the promoter. And then they gather the RNA polymerase says and say, okay, please make the copy here. So that's really important because it says that the promoter is the place that stimulates and differentiates one cell and one gene from another. This process of making an RNA copy of a gene due to the promoter and the specific nature of the promoter, and this will be important when we talk about biotechnology, is called gene transcription. The next stage is gene translation. So now we have an RNA copy at the ribosome, and we're going to translate that into amino acids and proteins. This is done by bringing the amino acids to the ribosome. And how is this going to be accomplished? It's going to be accomplished by another type of RNA called transfer RNA. Transfer RNA grabs the amino acid out there in the cell and brings it to the ribosome, and transfer RNA, it's made up of nucleotides, and the nucleotides in transfer RNA are opposite to those in messenger RNA. So when it sees AAA in messenger RNA, the transfer RNA will have the opposite nucleotides to that and bind to it. What is the language? The language is called the genetic code. And the code words are 3 bases each. Let us say the language was only 1 base. Let us say in messenger RNA, every time there was an A, you would bring in amino acid number 1. Makes sense. Every time there was a second, right? So there'd be 4 different nucleotides, 4 different amino acids. That's not going to work because you have 20 different amino acids. You need 20 different words in the messenger RNA to bring 20 different amino acids. Okay, there are 4 bases. Let's make 2 letters, right? AA, AG, GA, GC, right? There's 16 possibilities. That's not going to work either, right? We need at least 20 different words. And we have 20 different words if we have 64. So if there are 4 times 4 times 4 or 3 nucleotides in the code word in messenger RNA, we can essentially get the language that we need. The code was actually deciphered first by a scientist named Marshall Nirenberg in 1961. Nirenberg did a very famous experiment where he took a test tube and he added everything you need to make proteins except messenger RNA. And when he put in a messenger RNA that just contained U, it's the equivalent of T in messenger RNA, it's a different nucleotide called uracil. He put in a bunch of U's, and he asked which amino acid is going to be put into protein. Okay, so he had this in the test tube. He had all the amino acids there, and he said, which amino acid will be put into protein if we have the signal U in messenger RNA? And it turned out to be an amino acid called phenylalanine. Next, he tried a messenger RNA that had AAAA, and it got lysine. Then he tried the CCCCC, and he got another amino acid called proline. So that's the way he deciphered the code, with these artificial test tube experiments with messenger RNA. The sociology was very interesting. Nirenberg was a very young, not famous scientist, and he was not a self-promoting kind of person. He presented a talk at the International Congress of Biochemistry in 1961 in Moscow to an audience of less than 10 people. Everyone was going after what the genetic code was. What's the messenger RNA that makes that primes the synthesis of amino acid? And this guy that no one ever heard of had done it. It turned out that Crick of Watson and Crick was in the audience because he had seen the title of this thing and he said, wow, this guy may have cracked the genetic code. And Crick walked up to Nierenberg afterwards and said, "Would you like to give your talk again tomorrow morning on the last day of the conference?" And the guy said, "Oh, okay, I guess I'll you know, I was going to leave, but I guess I'll give a talk again tomorrow." There were 2,000 people in the audience, and they gave him a standing ovation. And a couple of years later, he won the Nobel Prize. The genetic code is a very fascinating thing. There are code words for all 20 amino acids. And these are determined, again, in the messenger RNA that comes from the DNA. That's the most important thing genetically that I want you to know. The DNA makes a messenger RNA that has the code word that determines the order of the amino acids. And the code is in the messenger RNA. The code is virtually universal for all life on Earth. Amazing. We use the same genetic code as coconuts, as bacteria. We have a common language. This implies that we're all related in some way, right? And evolutionarily, we'd call that descent with modification. It takes about a minute for a cell to make a protein with 500 amino acids. Things are happening pretty fast down there. Antibiotics, such as tetracycline, for example, block the ribosome in prokaryotic cells, like bacteria that infect us only, because the ribosome has slightly different components in prokaryotes as it does in eukaryotes, such as plants and animals. So these antibiotics can take advantage of that by binding to that particular ribosome. Now you might ask, well, how do I become resistant to an antibiotic? And obviously, the gene, right, coding for that particular protein that's in the ribosome to which the antibiotic binds might mutate. The protein in the ribosome will fold differently. The antibiotic can't bind anymore, and now you have antibiotic resistance. We'll talk about that later on in the course. Genetic coding explains the phenomenon of mutation. Consider phenylketonuria, a disease I've talked about several times in the course. It's genetically inherited. A gene codes for a defective enzyme when it's in a mutant form called phenylalanine hydroxylase. Remember, phenylalanine is converted to tyrosine, and when this doesn't happen, mental retardation can be the result. Let's now define this in the precise terms that we're able to do at this point in the course. In the normal situation, in the dominant gene that codes for the protein phenylalanine hydroxylase has 451 amino acids. In the most common mutant form, in phenylketonuria, the protein has 451 amino acids, same length. At position number 408 of the 451, the coding information in DNA is CGG in the normal situation. At position number 408 in the mutant situation, the coding information is TGG, a single base, a C in the normal, a T in the mutant. Now we're beginning to define what a mutation really is. It's a change in DNA, right? A single letter has changed. The amino acid that is prime from this in the chain at position 408 of 451 is called, I'll give you the name, arginine in the normal and tryptophan in the mutant. Now, the arginine at position 408 causes the protein to fold in a certain way, and it catalyzes the reaction of phenylalanine converting to tyrosine. All is well. That single amino acid change to tryptophan, one out of 451 amino acids, causes the protein to fold abnormally. So now it doesn't catalyze the reaction going from phenylalanine to tyrosine, and all of the clinical results happen. A single base pair change, a C to a T, out of 3 billion in the genome causes all of the clinical characteristics of this disease. So now we can define a mutation. A genetic mutation is defined chemically as a change in a single or multiple bases in DNA. A C going to a T, for example. And then we can start talking about mutagens and what they do. We can define genetic capacity, the ability to produce a phenotype now, as the presence of DNA sequences that code for proteins with specific functions. And now we know what coding information is. We now turn in the next lecture to the genome, where we look at the total genetic sequence and the capacity of an organism. Thank you.